afternoon to all of you. As he has already introduced me, I am Bharti Choudhary and I am doing my PhD from Guru Gobind Singh International University, Delhi, India. I am going to present my research work on Antagonist Streptomyces exfoliatus MT9 as biocontrol agent against fruit-rotting fungi. Now, I would like to mention one thing, that I have not given the experimental details over here, so if you have any query regarding the experimental section, you can ask me later. Now, first of all, why there is a need for biological control? The fungal spoilage accounts for a significant level of post-harvest losses of fruits and vegetables, and it has been estimated that about 20 to 25% of the harvested fruits are decayed by pathogens during the post-harvest conditions. And in India specifically, according to the Central Institute of Post-Harvest and Engineering and Technology, it has been estimated between 5.8 to 18% that accounts for 2 lakh crore annually, that is approximately $3 billion, that due to the inadequate cold storage facilities and lack of proper food processing units. The need for biological control is due to the negative impact of synthetic fungicides and pesticides in food chain on human health and environment, and the requirement is more so that there could be more integrated approaches to reduce the chemical residues in the food chain. Now, this slide shows the citrus fruits which are decayed by different fungi such as Penicillium digitatum, Penicillium italicum, Phytophthora species, Geotrichum candidum, Colatotrichum gliosporoides, and Botrytis diplodia theobrome. However, the major diseases that occur in the citrus fruits are caused by green mole, blue mole, and sour rot, which is caused by the Geotrichum candidum. In case of papaya, the major disease is caused by Colatotrichum gliosporoides that leads to the anthracnose lesion, and it has also been affected by fungi species Phomopsis caricae papaya and Rhizopus stolonifer that causes the Rhizopus rot. Now, the synthetic fungicides such as imazalil, thiabendazole, benamoil, and carbendazim are the primary means to control the post-harvest decay of fruits in storage conditions. However, the application of these synthetic fungicides to the harvested commodity has been curtailed by the development of pathogen resistance to key fungicides, the lack of replacement fungicides, and the negative perception regarding these synthetic fungicides. Therefore, there is a strong public and scientific desire to seek for safer and eco-friendly biocontrol alternatives, and hence biocontrol can be exploited as an effective method for controlling the post-harvest decay of fruits. Biocontrol can be defined as the use of naturally occurring microorganisms to antagonize the post-harvest fungal pathogens. It exploits the natural cycles with reduced environmental impact, and it fits the sustainable agriculture. Therefore, with the aim of developing a biocontrol agent, first of all, we isolated the actinomycetes from the soil of Loktak Lake of Manipur, India, which is a very popular lake in India, and on the basis of the morphological characteristic, we picked and streaked them onto the inorganic salt starch medium, that is ISP4 medium, and among them 4 actinomycetes isolates, that is MT7, MT9, MT12 and MT59 were selected for further antagonistic studies against the fruit-rotting fungi. Next, we performed the in vitro antagonism of these selected Streptomyces isolates against various fruit-rotting fungi by using the cross-plug assay, and it was observed that a strain MT9 showed good antagonism against most of the tested fungi. Since strain MT9 showed excellent antagonism, it was further screened against a variety of fruit-rotting fungi, and it showed a broad spectrum antifungal antagonism against all of the tested fruit-rotting fungi. Then we performed the cultural characteristics of strain MT9 for its identification on different culture media. We observed the growth, aerial mycelium, substrate mycelium, its sporulation and pigment production on different culture medium, and all these observations confirm that the strain MT9 belongs to the Streptomyces species. For further confirmation, we went for molecular characterization and phylogenetic analysis, and on the fatty acid methyl ester analysis of strain MT9, It showed a characteristic 17:0 cyclopeak, which comprised of 1.22% of the total fatty acids, and this 17:0 cyclopeak is a strong indicator of the presence of Streptomyces species. Then on the phylogenetic analysis, the blast analysis of 1364 nucleotides of 16S rRNA gene sequences of strain MT9 showed 100% similarity with the Streptomyces exfoliatus, confirming it to be of Streptomyces species. Now the main components of the fungal cell wall are chitin, glucan and protein. Therefore, the Streptomyces exfoliatus MT9 was screened for the production of these three fungal cell wall lytic enzymes on agar plates embedded with specific substrates, and a zone of clearance around the antagonist MT9 indicated the secretion of these three hydrolytic enzymes, that is chitinase, beta-1,3-glucanase and protease. We also performed the time course experiments related to the growth and hydrolytic enzymes production, and it was observed that the chitinase production was maximum on the 6th day of incubation, beta-1,3-glucanase production was maximum on the 5th day of production, and protease was found to be maximum on the 2nd day of incubation. Then we performed the in vitro antifungal activity of the extracellular metabolites present in the cell-free culture filtrate, and it showed good antagonism against most of the tested fungi, whereas the heat-treated and proteinase K-treated cell-free culture filtrate did not show any antagonism against the tested fungi, indicating that the nature of the extracellular metabolite was heat-labile protein. It was due to heat-labile proteins. Then we performed the mycolytic enzymes activities and stabilities as a function of temperature and pH. The optimum temperature for chitinase, beta-1,3-glucanase and protease was found to be 50, 60 and 70 degrees, whereas the thermostability was maximum for chitinase at 50 degrees, for beta-1,3-glucanase at 50 degrees, and for protease it was 40 degrees. The optimum pH for chitinase, beta-1,3-glucanase and protease was found to be 8, 5 and 9, and they were found to be stable at pH 8, 5 and 5 respectively. Now, in order to visualize the isoenzymes produced by the Streptomyces exfoliatus, the 8-day samples of crude cell-free culture filtrate were run on the SDS page with the specific substrates, and it was observed that the Streptomyces exfoliatus secreted 6 chitinase and isoenzymes, and 6 active bands were also observed in protease zymogram, indicating that the Streptomyces exfoliatus secreted 6 protease isoenzymes, and whereas an active band of beta-1,3-glucanase was observed on the zymogram. Next, we screen the Streptomyces exfoliatus MT9 for the production of antifungal secondary metabolite, and the Streptomyces exfoliatus was cultivated in M93 medium, that is glucose, yeast extract, malt extract, pH 7 at 30 degrees for up to 10 days, and each day sample was collected for determining the antifungal activity and the growth of the organism. And it was observed that on the 6th day of incubation, the antifungal activity was maximum when the growth was in the stationary phase. In addition, we also screened the organism for the siderophore production, and orange halo zone around the antagonist indicated the chelation of ferric ions from the intense blue colored complex to the siderophore, which is produced by the Streptomyces exfoliatus MT9. Then we checked the in vitro antagonistic potential of extracellular culture fluid, and it showed broad spectrum antifungal activity against most of the tested fruit-rotting fungi. Now, in order to isolate the antifungal secondary metabolite from the extracellular culture fluid, it was sequentially extracted with different organic solvents ranging from N-hexane to N-butanol, and N-butanol exhibited maximum antifungal activity against all of the tested fungi, whereas N-hexane diethyl ether chloroform and ethyl acetate showed less antagonism. Since N-butanol extract was found to be the best solvent for the extraction of the antifungal secondary metabolite, this time we directly extracted the extracellular culture fluid with the N-butanol in 1:1 ratio, and it was tested for its antagonism, and it showed a broad spectrum antagonism against all of the tested fungi. Then we determined the minimum inhibitory concentration of the antifungal compound in the N-butanol extract by using the poison food technique, and the MIC was found to be 2 microgram per ml for Colatotrichum gliosporoides. It was 2 microgram per ml for Geotrichum candidum, 5 microgram per ml for Rhizopus stolonifer, and 25 microgram per ml for Aspergillus niger, whereas the 100% inhibition was found to be at 1.6 mg per ml for all the tested fungi. Now, to determine the number of antifungal components present in the crude N-butanol extract, we also performed the TLC bioautogram, in which the crude N-butanol extract was run onto the TLC, and the TLC was then overlaid with the PDA medium containing the Geotrichum candidum spores, and it was incubated for up to 24 to 48 hours. And after 48 hours of incubation, a single zone of inhibition was observed onto the TLC bioautogram, indicating that a single antifungal component was present in the N-butanol extract. Then, in order to characterize the antifungal component in the N-butanol extract, we performed the ergostrol inhibition assay. Ergostrol are basically the main targets of the polyanic antifungal compounds. So in this experiment, we have tested the N-butanol crude extract onto the PDA plate supplemented with 0.5% ergostrol, and as we can see that in the presence of ergostrol, with ergostrol, there was a reduction in the zone of size of zone of inhibition as compared to the plate in which there is no ergostrol, and this indicated that the nature of the antifungal compound is polyene. We further confounded by the antibacterial activity, and in this the N-butanol crude extract was checked against the E. coli and Bacillus subtilis, and it did not show any antibacterial activity as the bacteria do not have sterols in their cell wall. Next, we also did the UV spectrum of the N-butanol crude extract, and the absorption spectrum showed a characteristic maximum absorbance peak at 224 nanometer that fall in the UV range, and which is a characteristic of the polyene antibiotics. Next, we perform the in vivo bioassays to determine the efficacy of the different biocontrol agents, that is cells of M93 broth, extracellular culture fluid, its N-butanol extract, cells of chitin medium, cell-free culture filtrate, and a chemical fungicide, that is carbendazim, as compared to the control. And as it is evident from the image that the biocontrol agents displayed a better disease suppressive activity than the chemical fungicide as compared to the control. Next, we calculated the disease incidence in oranges on 3rd, 5th and 10th day of incubation. On 10th day of incubation, it was observed that the disease incidence was reduced to 0% in case of cells of M93 broth, its N-butanol extract and cells of chitin medium, whereas the disease incidence was reduced to 20 and 25% in extracellular culture fluid and cell-free culture fluid, whereas the carbendazim, that is the chemical fungicide, reduced the disease incidence to 55% only as compared to control. We also calculated the disease severity in oranges on the basis of an empirical scale, and on the 10th day of incubation, it was observed that a disease severity reduced to 0% in case of the cells of M93 broth, its N-butanol extract and the cells of chitin medium, whereas the ECF and CCF reduced the disease severity to 8% respectively, and the carbendazim reduced the disease severity to only 23% as compared to the control. Therefore, on the basis of all these results, we can conclude that the Streptomyces exfoliatus MT9 and its extracellular metabolites can be employed as a effective, newer biofungicide for controlling the post-harvest decay of fruits in storage houses. From this, I would like to thank my professor, my supervisor, Professor Rajendra Kumar Gupta, my senior Dr. Anand Nagpure for his constant support in this in carrying out this research. I'm also thankful to University Grants Commission for providing me with the financial support in carrying out this research, and also to Department of Science and Technology for providing me the travel grant to come over here and present my work. And thank you very much for your kind attention. Have a fruitful day. Ask me questions.
Any questions? Yes?
Yes, ma'am.
Well, it seems to be quite effective from the image.
From the image.
I was just wondering, what are the costs to generate
the decay loss?
Yeah.
Decay loss is, sorry, can you repeat it?
I was just wondering, what are the costs for generating this extract? Like whether they balance.
We have not calculated the estimated the cost actually, but as I have done all these works in the laboratory, and it takes around 3 days to cultivate the culture, and then it takes around 6 days for the production of this antifungal metabolite. And nowadays I have been working on the optimization of the medium so that we can reduce the number of the days which it takes to produce the metabolite.
I guess it will be decreasing.
It is decreasing. Now I am getting the metabolite production maximum on the 3rd day itself.
Okay. Another question I have is, it seems to be very effective in curing the fungus. Because the fruit are going to be consumed by human later on, do you know if they have any cytotoxic effect?
No, we have not done that yet, but these organisms, as they have been isolated from the soil, they are the natural organisms, and they have been tested that they are not pathogenic to the humans. They have been tested that these are not pathogenic isolates. So we can use them, and what we can do in the storage, that we can coat them, and I have just specifically chosen the citrus fruits and papaya because we never eat them with the peel as such. We always peel them out while eating. So we can use this procedure for, and later on we can also do the cytotoxic studies and in clinical trials.
Any more questions? Yes?
Okay. From the dendrogram picture you showed, how many groups are we able to?
Sorry, ma'am?
How many groups?
That picture?
Yeah, from the dendrogram.
This one?
No. The molecular
The phylogenetic tree.
phylogenetic. What information did you get from that?
In this, we have done the blast analysis, basic local alignment search tool that we have used, and in this we have on the basis of the blast, we pick the top 30 hits which matches with the organism. Top 30 sequences that matches with the organism, and the E value, which is the expected value, and it shows that if the E value is closer to the 0, then there is a high probability that it is a similar organism.
So that means everything you got was the same organism.
Yeah, Streptomyces exfoliatus.
Since E was 0.
But the species is different.
Yes? I have one question.
Yes.
Besides microbiologists lying to going to exotic places, why do you think soil samples from that lake would be good to investigate?
I have chosen that soil only because that lake has been unexplored till now. There have been no reports as such previously that from that the actinomycetes have been isolated previously, and on the basis of the literature that these organisms have not been tested against these fruit-rotting fungi, and that area is specifically very less explored. That I have taken it from the floating vegetation. The soil has been taken from the floating vegetation from this Manipur lake, Loktak lake.
And what about identifying the active ingredient in the in-butanol?
That I'm doing.
Okay.
Yeah, I'm going for the further purification and the identification by GCMS and LCMS. So that I'm doing.
Okay. Best of luck.
Thank you for that. Yeah.
Oh, yes, please.
Yes. The treatment was
only in the trees, right? And what happens with the trees? Because in some cases the disease are in the trees or in the plants, and then
it is transferred to the fruits.
Yeah.
Actually, these fungi which I have targeted, they only are able to propagate after the post-harvest. After you have taken out them from the fruit, from the tree, then only these fungi are capable of attacking those fruits. So I have specifically taken those fungi only which are propagating in the post-harvest conditions. So the fungi which basically attacks the tree itself, they are different from these fungi. I hope it satisfies her. Thank you.